So, good morning, good evening, and welcome to all of you to PaleoPerks. And today our speaker is Pablo Milla Carmona from the University of Bristol in UK. And he will be telling us about the transition between micro and macroevolution from a morphological perspective. So this talk has been financially supported by the Linnean Society, and we thank them for providing financial help for this talk. And this is the general format of today's seminar. At first, we have welcome and announcements, which is currently going on. And after this, we'll have the talk for approximately 30 minutes. Following the talk, we'll have a moderated question answer session for 10 minutes. And after that, we'll have the tea time session, which Pablo has very kindly agreed to stay to. And if you have any questions, please send them to questions@PaleoPerks host. And Rehmat is our questions host for the day. And after the session, if you want to ask any questions directly, you can use the raise your hand button. A bit of housekeeping. So at PaleoPerks, we value the participation of all folks who are interested in paleosciences. And please remember to abide by our code of conduct. Please mute yourself for the duration of the talk. And like I said, you can send your questions to questions@PaleoPerks host or using the raise your hand function. Now we have the closed captions inbuilt in Zoom. You can use the CC button to show or hide them. And if you are a early career researcher, or if you know somebody who is doing good work, please don't hesitate from nominating themselves or yourself. And this is the link. We also have a weekly feedback form wherein we encourage you to provide your comments and your feedback so that we can get to know what we can improve on. And also we are looking for new committee members. So please contribute and support this initiative. We are looking for speakers who are working in different aspects of paleosciences, and the applications are due in October 31st. The application link will be sent in the chat. And so now I would like to introduce our speaker, Pablo Milla Carmona. He did his BSc from the University of Buenos Aires, and he obtained his PhD in geological sciences from the University of Buenos Aires. And currently, he is working as a postdoctoral researcher in the University of Bristol in UK. And with this, I would like Pablo to start his talk, and the floor is yours.
Okay, thanks for the introduction. Are you able to see my my presentation in presentation mode?
Yeah, it is visible.
Okay, perfect, thanks. So that's right, I am Pablo, and I am going to be talking about how I study the transition between a micro and macroevolution from a morphological perspective. But first, to complement a little bit, just a few words so you can understand where I'm coming from. I am originally from Chile, but I left basically because I wanted to study paleontology, and Chile at the time didn't have any degree in paleontology. So I basically went to the closest place where I could study paleontology, and that was the place was Buenos Aires in Argentina. And I just want to stress the great public education system that Argentina has, that allowed me to, and the public scientific system as well, that allowed me to had access to a great education, a public education for free. So I will be forever in debt to the people from Argentina for basically supporting my research. I did my bachelor and my PhD and one postdoc at the University of Buenos Aires, financed by the CONICET from Argentina. Although I am currently a postdoc at the University of Bristol. This is a position I just took, so that's where I'm going to be the next couple of years. Then again, with the goal of you understanding where I'm coming from, one of my intellectual heroes was Stephen Jay Gould. So I was kind of fixed. Some people would say I was obsessed with studying macroevolution from a paleontological perspective, and that usually entails working with morphology. So this has been the main theme of all my research, and something that came to fruition during my PhD, during which I studied the anagenetic patterns of evolution. This is just a fancy way to say I tracked changes of different lineages through time. I did this in 13 species level lineages belonging to these 3 genera of fossil bivalves from the lower Cretaceous of Argentina. I did so over a time interval of roughly 7 million years, for which there is an excellent time control, thanks to several decades of biostratigraphic and also more recent geochronological research. Well, the reason I choose to work with these 3 genera is that they are actually a very nice model, because they are phylogenetically diverse. They come from different parts of the bivalve tree of life, but they also share the same basic lifestyle. They are marine shallow burrowers, and they can be found in roughly the same paleoenvironments. So they are at some level paleoecologically comparable. And of course, also because there are tons of fossils of this genera, so we could do some statistics with them. Here, I'm showing you the results I got for Ticomia, which is the first of the genera I studied during my PhD. And the example I used to develop my work pipelines, so to speak. And this pipeline basically involved first devising a strategy to quantify several aspects of the morphological variation in shell morphology of these bivalves, including complex traits like ornamentation patterns. I did so using geometric morphometrics. Then I tried to figure out how many morphologically discrete groups there were in the sample of each one of these genera. And finally, I reconstructed the stratophenetic trajectories. This is change through time, through stratigraphic levels for each one of these groups. And the results I found for Ticomia, sorry, are very much the same I found for all the other species I studied. And this is that although there is some room for directional evolution, mainly associated to gradual changes in the paleoenvironment, actually most of the lineages are under stasis or evolving under a random walk model. And well, you can see that there is actually very the morphologies of these lineages are basically stable, and they could also be fluctuating randomly. And this was the main pattern I found. By the end of my PhD, I was trying to make sense of these results in order to gain some more general evolutionary insight. And I came across this work, this paper from Ullera and colleagues from 2011, which contained the unmodified version of this figure, which is showing us more than 8,000 points. Each point represents an amount of morphological divergence that has accumulated on a particular time interval. So in the y-axis you have accumulated divergence, and in the x-axis you have the time it took for that divergence to accumulate. And you can see that the pattern is very clear. Below the 1 million years mark, you can see that there is very little or known accumulation of divergence at all. And then above, starting from the 1 million year mark, you can see that divergence start accumulating very slowly, and then above the 10 million years mark, you can see that accumulation of divergence basically explodes, right? It amplifies a lot. And so this figure had me thinking, because we know that this part of this figure is representing time intervals that are usually involved in populational studies that address microevolutionary phenomena that we can directly observe and study and record, while the other end of this figure is representing time intervals that are usually at play for studies framed within a phylogenetic framework. So we have many studies in this end of the spectrum, and there's then a lot of studies at this end of the spectrum. But in order to understand how we go from here with this very insignificant accumulation of divergence to here, where divergence is accumulating very rapidly, we need to understand what's happening in these intermediate time intervals, right? We need to understand how the microevolutionary phenomena that we know are operating within the species scale up into the macroevolutionary variation that is then the target of more general macroevolutionary studies. And so during the rest of this talk, I'm going to be addressing two cases I had the opportunity to work with after my PhD, in which I study small systems, and I do so by trying to relate intraspecific and interspecific morphological variation. The first of the cases I'm going to address is the evolution of ontogeny in the genus Steinmanella. Steinmanella is a genus of trigonid bivalves. This genus was other of the genera I studied during my PhD. In this case, we had 7 species distributed over this 7 million years time interval, but the results were the same. As I said, widespread stasis was the most common pattern here. But we were especially interested in this genus for the same reason it is very conspicuous, and that is because of this very eye-catching morphology. You can see the different species have a very interesting geometries, and you can see also they have these complex patterns of ornamentations that suggest morphological models and what have you. So we were particularly interested in the ontogeny of this species, since ontogeny is a fairly important paleobiological property of extinct taxa, and also because an additional advantage of working with fossil bivalves is that you have a record of the shape changes that happened during the life of the animal, because all these changes get recorded in the growth lines in the shell, in the outer surface of the shell. So we just collected some longitudinal data from almost 70 specimens belonging to these 7 species. So we could reconstruct the ontogenetic trajectories of each one of these species, and we could compare them within the genus. We did so, and then quantified shape variation using 3-dimensional geometric morphometrics. In this case, we used curve and surface three-dimensional semi-landmarks. And then we analyzed the data of each one of the species using multivariate segmented regressions. We tried with different number of segments and then applied model selection tools to find out which one of these models was the best fit for our data. And that's how we found that all the 7 species, the best fitting model was a regression with 2 segments for all of them, with a single breakpoint. This is what you are seeing here, all the trajectories of all the 7 species projected into morphospace. And you can see that trajectories start advancing in one direction through the morphospace, and then they break and start advancing in a different direction. We also saw that dispersion around the ontogenetic trajectories, of each species was higher at the beginning of the ontogeny. This is size, and you have here the percrusted distances between all the shapes we measured and the expected shape under the ontogenetic trajectory. And you can see that these distances have a negative slope. So basically, as the animal grow larger, they are more and more packed around the ontogenetic trajectory within each species. Then we also projected the transformations implied by each one of the phases we found of all the species. This probably requires a little bit more explanation. Much in the same way that different points in a morphospace represent different shapes, different points in a transformation space represent different transformations that can be applied to any shape. So we just projected here the transformations implied by each one of the phases. And to our surprise, these transformations are fairly structured. We can see that we have all the phase 1 of all the species grouped together in this part of the plot. And we also have all the second phases of all the species grouped together here in this part of the transformation space. So whatever the changes are, they seem to be very conserved within the genus. Then we just calculated the average transformation of each group and then amplified transformation by 6 and applied transformation to the average shape in the sample. And that's how we were able to see that the first phase entailed or can be described as a crescentic mode of change. This means that as the animal becomes larger, the shape of the shell becomes more and more banana-like. And then we have a much more simpler second phase of anteroposterior elongation that is very common in bivalves. And finally, in order to get some evolutionary insight for this case, we also performed a series of ancestor-descendant comparisons. Basically, we compared the ontogenetic trajectories of pairs of species that are supposed to represent ancestor and descendants. And by doing this, we saw that most of the changes, the evolutionary changes between trajectories, were concentrated early in ontogeny, concretely at the starting point of the trajectories. And then these evolutionary changes in trajectories become more scarcer as the ontogeny progresses. And so trying to combine all this information into an interpretation, our proposal is that actually there is a an interval early in life during the Stemanella ontogeny, in which shell shape is much more plastic. This is evidenced by the crescentic mode of change, which is much more complex and demands more flexible patterns of secretion by the mantle. But then this initial phase of complexity fades away as the more simpler antero-posterior elongation trend takes over. And well, this is consistent with both the higher dispersion at early ontogeny that we observe within each species, and also is consistent with the fact that we have more interspecific differences in trajectories early at ontogeny. And this would be basically because changes early in ontogeny are more likely to result in more substantial morphological change. And so changes happening early in the ontogenetic trajectory are much more likely to result in a cladogenetic event. That's all. On the Stemanella front, we are also exploring patterns of modularity and other interesting stuff, but that's yet not ready. So I'm going to now talk about the second case of this talk, which is the study of convergent evolution of exaggerated tail morphologies in Tyrannus. So here we worked with Tyrannus, which is a genus of passerine birds that originated roughly 5 million years ago. It comprises 13 species distributed across North, Central, and South America. Most of these species have what we could call normal or regular tail morphologies. But we also have 2 species of independently evolved flycatchers, the scissor tail flycatcher and the fork tail flycatcher. And both these species have independently evolved these very conspicuous tail morphologies in which the outer feathers of the tail are very, very elongated. So we know that that bird tails are under different selective pressures. The 2 most important usually being sexual selection operating on variability that individuals show regarding their capacity to attract mates, and also more standard natural selection operating on variation on aerodynamic performance. This is the tail of the birds understood as a flying device. But we were actually more interested in the structure of the underlying variation that is available for this process. So we could learn a little bit more about the role that this structure has on the recurrent evolutionary origin of these very particular tail morphologies. So this time we quantified morphology of tail shape using 2-dimensional landmarks. We put some at the base of the tail and then some more at the tips of the feathers of the tail. We did so for 280 specimens. So we had a reasonable amount of specimens per species, roughly an average of 20 specimens per species. And we also had the phylogenetic relationships between these species that we took from the work of Harvey and colleagues in 2020. And this is how variation in tail shape in Tyrannus looks like. You can see that species with normal tails are confined to this small region of the morphospace sorry, and are also heavily overlapped. And you can also see that the flycatcher species are highly divergent, and they occupy this portion of the morphospace representing deep forked tails. But nothing of this was unexpected. We knew that something like this should arise when quantifying morphology in these groups. But since we were interested in dissecting intraspecific variation in order to relate their sources to evolutionary change, we also computed a few axes of a few shape axes capturing different aspects of morphological variation. We first calculated an axis of maximum interspecific variation. We did so only for species with regular tails. This was meant to capture the regular within clade dynamics within Tyrannus. And we also, for all the 13 species, including flycatchers, we also estimated two axes of intraspecific variation, an axis of sexual dimorphism, basically reflecting the transformation that happens between the typical male morphology and the typical female morphology. And then another axis for allometric variation. This is just shape transformations that are associated to changes in size. And when we project this axis into morphospace, we can see that Tyrannus species with normal tails are actually very variable in both the length and the orientation of this intraspecific axis. And actually, there is no obvious correlation with the maximum interspecific axis of variation that, remember, was computed using only this species. On the other hand, we can see that flycatcher species are fairly consistent in both the length and the orientation of the trajectories. And actually, these trajectories are kind of parallel to the axis of maximum interspecific variation. So in order to confirm the patterns that we were seeing, we performed a series of trajectory analysis. And this basically confirmed that species of Tyrannus with normal tails had both shorter axis of intraspecific variation, meaning that the transformations were more subtle, and crucially, they were, well, not statistically correlated or aligned to this axis of maximum interspecific variation. While for flycatchers, we had the opposite pattern. Both intraspecific axes were longer, meaning that the transformations were more marked. And more importantly, both intraspecific axes are statistically aligned to this axis of maximum interspecific variation, sorry. And so our interpretation of this case, and I hope this makes sense, is that actually this axis of interspecific variation is capturing a direction of evolution or a component of morphological variation that is preferentially followed during cladogenetic events within Tyrannus. So whatever the processes that are behind this, the emergence of this interspecific axis of variation, this represents kind of an open lane of variation for macroevolutionary variation. But also, to get here, in order to obtain this extreme divergent morphology of flycatchers, we need a combination of several factors. First, we need a process of sexual selection that we know is a process, a very powerful process that can produce substantial morphological change, that is being fueled by allometric variation, which is another biologically important source of variation, usually ecologically significant and pervasive. And not only that, but this process is actually driving mean morphology through this open lane of macroevolutionary variation. So that's how, or that would be how the divergence gets amplified, right? So we already knew that there was a process of sexual selection going on in this species, but the new insight we got here is that we also need a combination of several factors, including allometric variation and biases in the production of macroevolutionary variation. Well, that's it for the second example. Now trying to wrap up a little bit these the results or the insights we got from these two cases. I think these are very nice examples in the sense that they show how much can we learn from this study of intraspecific and interspecific variation in these small systems with few species that evolved within this 1 to 10 million years interval. Both studies addressed structural properties of variation. The Stemanella case delved into ontogenetic variation, while the second case was more about allometric and macroevolutionary lines of least resistance. And I think both show that studying structural properties is important, and we should strive to do that if we are going to understand evolution fully. But also, both cases are lacking the functional side of the story. This is, we lack yet for these examples, an idea of the adaptive value of morphological variation. So that should be the aim for the future in both cases, to measure functional diversity in order to see if, or in order to measure the relative contribution of structural and functional properties to the evolution of these systems. Admittedly, I am functional analysis is not something I have a lot of experience working with. But as I mentioned at the beginning of this talk, I am now working at the University of Bristol with the paleobiology group. And that group is full of people who are very, very good at functional analysis. So I'm very excited about what I'm going to learn next in the next few years, and what I'm going to be able to apply to those cases. But for the time being, I have been focusing on developing Morphospace, which is an R package for building and depicting multivariate ordinations of geometric morphometrics data. So everything I have learned so far, both in my PhD and my postdoc research, is being poured into this package, including this logic of thinking about evolutionary processes as objects into Morphospace, or the tools I use to get there, and my work pipeline. All that is being translated into Morphospace, which is, as I said, specifically devoted to work to build and visualize morphospaces. For all the other fun statistical analysis, you have great R packages. Morphospace is not going to be useful for that, but these are the kind of visualizations that the package can produce, and I hope this can serve researchers to explore their data, sharpen their hypotheses, and publish their stuff. So here you have the GitHub repository in which the package currently lives. So if anybody thinks it can be useful. And yeah, that's all from me. I want to thank you all for coming, for your attention. I also want to thank the different institutions that have supported my research. And of course, special thanks to the Paleo Perks committee for inviting me to take part on these seminars. Thank you all very much.
Thank you so much for that excellent talk, Pablo. We would request you to keep your last slide up. And I will start putting in the questions in the chat box. Yeah. Yeah, the first question is from K. Sendor Sarwan, and he wants to know, for any sexually dimorphic taxons or a taxon which exhibits any form of polymorphism, would each form or phenotype undergo stasis through time, and how would divergences affect a highly polymorphic population?
Would each form? I don't think there is a unique answer to that. I think it depends on the case. I'm no expert on sexual dimorphism, but I guess it would be interesting in each case to check whether the different shapes corresponding to males and females are actually stable or are evolving as well.
Okay. The next question is also from Sender, and his question is, you mentioned that changes in a juvenile stage would be more likely to result in a cladogenetic event if the organism or population was in a pedomorphocline during a given time period. Is it plausible to expect more speciation events and morphological divergence in this period too?
I think so. That's a very interesting observation I hadn't thought about before, but I think the answer is yes. If we have pedomorphosis and the entire ontogeny is becoming shorter or more juvenile, we should expect this period or this interval of more shell shape plasticity to result in a higher amount of speciation events. That's what I would expect, definitely.
Okay. Okay. So, yeah. Yeah, the next question is, how would biotic homogenization induced by environmental stresses affect the mean interspecific morphological variance, and would it bottleneck the evolutionary lane or trajectory for a population or species?
Trying to understand the question. Biotic homogenization. I don't know what, I'm not familiar with the concept of biotic homogenization. But I guess if you have some sort of homogenization, you should expect well, morphological variants to get contracted, right? So morphological variants. Oh, and the last part of the question, if it would bottleneck the evolutionary lane or trajectory. I guess so. I guess the extent of the evolutionary of least resistance will be dependent on the actual amount of divergence that can be achieved in single speciation events. That's what I think, in principle at least. So yeah, the amount of divergence should be dependent as well on this kind of homogenization processes.
So biotic homogenization is the process by which apparently two or more spatially distributed ecological communities become increasingly more and more similar over time. So kind of like a sort of a character convergence. So I think, yeah. So I think the question is, maybe does some sort of a convergence affect the variance or of that sort?
Well, I guess so. These evolutionary lines of least resistance can, I guess, function in both ways, like amplifying divergence, but also predicting where convergence can happen. In fact, you can see that the particular morphology of flycatchers is largely defined by the direction of the evolutionary lines of this resistance, right?
Right.
I don't know if that answers the question.
Yeah. Okay. The next question is, smaller specimens are more easily damaged or affected by diagenesis. Could this affect your observation of higher variance in early ontogenetic stages in bivalves?
Oh yeah, sure, of course. Any source of morphological variation can affect this kind of inferences. I usually clean up all the different sources of variation I'm not interested in, or that can obscure the patterns we are seeing. So for example, for the example of Steamonella, I removed analytically all the differences that were due to biostratigraphic level or geographic provenance or inter-individual differences too. But in the case of preservation, I just try to use only specimens that I was sure were well preserved. Because if not, of course, the morphological variance would be inflated.
Okay. Apparently, so these were the questions from the audience. And before we go into tea time, let me just take over the screen sharing from you. Just a minute. Yeah. So thank you all for joining us. And we would now go in for our tea time. And before tea time, the question of this week is, what is a cool morphological feature that you would like to have from the animal world? So yeah, just walk around, move around, and take a break for 2 minutes, and then we'll see each other again.